have been talking about the cell cycle control, the protein network, the cyclin CDKs controlling that. But of course, the cell cycle control without the chromatin remodeling is pointless, because anyway, it's mostly about the DNA compaction, DNA segregation. So the main control is going through DNA, of course. So we should see how it's also controlled. Now we know that in the G1 phase, there is a DNA double helix. In the S phase, this DNA double helix is replicated. In the G2 phase, you have two DNA double helix, and these two DNA double helix, they are created by the DNA polymerase enzymes in the S cycle, in the S phase, so we know that they are identical. And how we call them? Sister chromatids. They are so identical, we even don't count them as two different chromosomes, we count them as one single chromosome. So here, starting from the S phase all through the G2 phase, the cell actually has twofold of DNA. But this twofold of DNA doesn't behave as if it is twofold, as if everything is normal, the transcription and translation level continues normally. Now, of course, these sister chromatids, they should be kept together, because if you lose them, let's think about the nucleus. One can go in somewhere in the nucleus, one can go somewhere else in the nucleus, and they will lose each other. This is something we don't want. This is why these proteins are kept, these chromatids are kept to stick to each other with the help of cohesin proteins. Cohesins are ring proteins, and they are keeping the sister chromatids together. Now, in the G2 phase, everything is all right, but starting with the M phase, with the prophase, things start to change, because in the prophase, this spaghetti-like relaxed chromosome will start to condense, and finally, in the metaphase, it is going to create this very condensed chromosome. So these cohesin proteins, they will be broken. They will be gone here, here. But if you break all the cohesin proteins, of course, at the end, the two sister chromatids will be separated. So not all of them will be broken. Some of them here at the centromere will be kept. Okay? These centromeric cohesins, they are going to keep the sister chromatids together, and this is actually what you see here. Here, although the cohesin proteins here are broken, otherwise it wouldn't be able to condense separately, because this is one chromosome and this is the other chromosome. With the condensing rings, by the way, they are very similarly structured rings, cohesins and condensins. Here the condensins are concentrated, this DNA, and here in the centromere, they are still kept together with the help of the last cohesin rings available. Okay, they are going to be separated at the M phase, at the end of metaphase, and this is also under control of APC, but I'm going to come back to this later. Just keep in mind this cohesin and condensing story. Okay, let's start the M phase. I'm pretty sure that you remember the M phase. Okay, here it is quite out of proportion. Normally interphase is much longer than the M phase, and M phase normally just takes 1 hour. But here, to show the prophase, metaphase, prometaphase, metaphase, anaphase, telophase, and cytokinesis is kind of amplified. All through this M phase, two cytoskeletal elements are highly used. One is the microtubule, and the other is actin. So with the help of these two with the help of these two cytoskeletal elements, the M phase here breaks the cell into two, so everything is replicated still, but they have to be divided equally. This equal division is done with the help of these two cytoskeletal elements. The intermediate filaments here are not used because, as you remember, the intermediate filaments are not dynamic like actin or microtubule. They are not highly reorganized like these two, and they are not, how to say, there are not proteins that is reorganizing them regularly. They are not dissociating and associating again. They are quite stable. This is why intermediate filaments, although they are very strong and they are very useful in every cell, they're not going to be used here in this thing. So here, the microtubules have a very specific jobs. Segregate the chromosomes equally. And actin has another very specific job. Separate the cytoplasm. Okay, two different jobs, two different cytoskeletal elements. Let's start with the microtubule a little bit. You know that all the microtubules, they are originated from center of the cell, which is called the centrosome, which is a pair of centrioles. So in the G2 phase, so not in the M phase, in the late G2 phase, the centrosome is already replicated. But after they are replicated, they don't go and leave each other. They stay together. Only and only in the prophase, they started to move. Now, in the cell, before the, well, let's say in the G2 phase, there's twofold of DNA already in the nucleus. The cell is big. It's actually waiting for a division. And there is the centrosome. It's actually now two pairs of centrioles. But it didn't establish the spindle apparatus yet. Okay? It has the centrosomes. But the centrosomes, which are microtubule organizing center, has to be broken, and instead it should make the spindle apparatus. So they are both made of microtubule organizing center plus the microtubules. The difference between a classical centrosome in the S or G1 or G2 phase and a spindle apparatus here is that spindle apparatus is more dynamic and it is moving. That is the unique difference. But to create that, in the early prophase, all the microtubules in the cell, they go under catastrophe. Okay? So these microtubules, they are depolymerized. And because they depolymerized, instead of this, you have lots and lots of microtubule subunits in the cell. And all these alpha and beta globulin subunits, they are going to be used to make the spindle apparatus. Everything will just be reorganized. This is why the M phase is so visible under the microscope. Everything is changing shape. Okay, here it has started to establish the spindle apparatus. So it's already duplicated in the G2 and in the M phase. These little microtubules, they started to grow this newly formed apparatus, and then they started to move to the opposite directions, and then they will catch the chromosomes. Okay, and you know that they hold the chromosomes from two different parts, and they are going to pull them as strong as they can. Okay, in the prophase, you know that the nuclear envelope is still here, it is still intact, and these poor microtubules, although they are growing, they cannot go catch the chromosomes because in between them there is the nuclear envelope. First, you have to get rid of the nuclear envelope. In the prometaphase, it is degraded, not really degraded, it is fragmented into vesicles. later they are going to come back together so that the microtubules can catch the chromosomes. Okay, at this point I should say that not all the microtubules of the spindle apparatus or catches chromosomes. First of all, some of them are growing the opposite direction because chromosomes are here in the equator, and these microtubules, they are growing in this direction. And of course, these microtubules cannot catch chromosome. There is no chromosome here. The chromosomes are here. These are called asters. Aster means star. They are star-like. And the function of aster is to attach to the plasma membrane. Here there is the plasma membrane. They are going to be fixed on the plasma membrane, and next they are going to pull the chromosome through the plasma membrane so that the cell is going to elongate. The second class of microtubules that you see here is the interpolar microtubules, or I should have a picture here. Yeah. These are the interpolar microtubules. They also did not catch a chromosome. This is just by chance. So there are chromosomes here. Some of them are lucky enough to catch a chromosome. It is just like fishing. So all the microtubules grow. They are like fishing. Some of them can catch a chromosome. Some of them they just catch each other. So this microtubule from here and this microtubule from here, they find each other and they become interpolar microtubules. This is important to establish the equator, because we know that when the cell divides, for the cytokinesis, we need this equator first. The chromosomes are going to align at the equator here second. Also, it fixes the structure. Third, there are going to be microtubules which are lucky enough that they are going to catch a chromosome from both sides. So there is a microtubule organizing center here, they come from this one. There is another microtubule organizing center here, they come from this one. They catch the chromosome and they pull to two different directions with equal forces. So it just goes, comes back, goes, comes back. So here, with this, like this game, so at the end they are going to be stabilized in the middle, which is called equator, and this is the metaphase. And this equator, this imaginary line, is called metaphase plate. Okay, here you can see the three different kind of microtubules. Biochemically or chemically, there is no single difference between these kind of microtubules. Just some of them attach themselves to the plasma membrane, some of them attach to the other microtubules, to other interpolar microtubules, and some of them to the chromosomes. Okay, next they pull, but of course this pulling is not successful because we know that there are still cohesion proteins here keeping things together. So before these cohesion proteins are broken, the microtubules will not be able to break them. Of course, these microtubules are applying a force for the big chromosomes, like the human chromosomes. There are like hundreds or dozens of them are pulling them, depending on the size of the chromosome. If you're talking about the yeast chromosome, even one single microtubule is enough. So, a strange fact, if you delete the microtubule organizing centers here, if you get rid of them, nothing happens. The chromosomes are not only passengers, they are actually quite active. The chromosomes can regulate the formation of the spindle apparatus, and the spindle apparatus forms anyway, of course without asters. So we are still not sure about the role of the centrosomes and the role of the centriole pairs. It's still a mystery of cell biology. Okay, and then next in the anaphase, the last the last cohesions are broken, and then the chromosomes, they start to move to opposite directions. which is what happens in the anaphase. Okay, let's continue with the